are interconnected, require you to take risks, and even though there's something about risk taking today that we're going to be talking about, but finding solutions that can be deployed, that can actually get out to address the need that has been identified. So that's kind of where we are as an institute, pulling together at the university, pulling together everybody across the university, as some of you already know, and if somebody on Zoom does not know, we are not in a department or a college. We are across the entire university, pulling things together. So one of the privileges of that I have is being able to introduce some of the best minds to our community, to share with them our story, but more importantly, learn from them, their story, and what they have to tell all of us. So Josiah is an assistant professor in the Department of Psychological Sciences right here in the University of Arkansas, and he directs what I was trying to figure out, what is it when I first met you? Full stack affective neuroscience lab. Affective, effective, was affective, But I met Josiah because this new building that some of you know, that big behemoth that's coming up, is going to have an MRI going into it, a 3 Tesla MRI. And who was the resident expert? Josiah. So we talked to Josiah about it, and we got informed more about him, but all the neuroscience community within the university. And then we did a long trip to Tulsa, to Laurent Institute, to see their system. So that was actually perhaps the first time where I got one-on-one time in the car for 2 hours, going out, coming back in, to be able to talk to Josiah and see how excited he is about what he's doing, and how he's excited not just about a narrow space of his own, but to think about a much broader picture and to be able to think about solutions. And he will tell you about what he's looking at in brain of what affects unfortunately an adolescent population that has needs, and that has fallen into a trap that we wish nobody falls. Josiah earned his PhD in psychology from Stanford. He did a postdoc in Indiana University in Bloomington, and very specifically, he looks at research using brain imaging of structural connections within the brain. And he is looking at adolescents who may have issues with their decision making in a way, and that aberrant or errant decision making leads some of them to drug use now or later in their lives, right? So with this in mind, let's welcome Josiah to today's talk.
Thank you. Well, thank you for the wonderful introduction, Ranu. I'm really happy here to speak with you all, and even people on Zoom, probably still in your pajamas at home. Just happy that we can all share this time together. I sort of selfishly wish that you had invited me to speak next year so that I'd be able to do this at the fancy new building, but that's no problem. We'll do it again next year, right? So a little bit about my lab. Like Ranu mentioned, we're on the main campus in Memorial Hall. And what's interesting is we sort of bookend the north end of the campus, whereas the new building, the new IQBR building, will be on Dixon Street, kind of bookending the south end of the campus. So psychology on the north, neuroscience on the south, and we'll cover all our bases. This is a lab, my lab. We presented some of this research that I'm going to present to you today at the Society for Neuroscience last year. And so my team, just a small crew of undergrads and grad students pursuing just full stack of affective neuroscience, and you'll get a flavor of what that means today. Right. Great, so these are the goals for today. Right, I'm going to try to answer 3 questions, and the first is I really want to teach you how we can actually measure the structure and function of reward circuits in any of you. All right, and MRI scans allow us to do this reliably, and once we have those reliable measurements, we want to deploy those measures to study hard questions or wicked problems, as IQBR would call it, in society. And one of those problems is drug addiction, but specifically, we have a couple of examples of how we can study stimulant drug addiction. So stimulants like cocaine and amphetamine and methamphetamine. These are dopamine agonists, and people get addicted to them, and we can study how that affects the brain circuits. And then lastly, another way to deploy these methods is to study, well, what are the antecedents? What precedes drug seeking at all? And so that opens up this question about using the methods to study adolescent development and maybe even predicting who will even try to seek drugs in the first place. All right. And so all of this, like Ranu mentioned, is really setting the stage for what's really exciting. And I've patiently waited 3+ years, and many of you have waited 10, 20+ years for a neuroscience facility here. But we are having a magnet at IQDAR, and hopefully open in a year and a half. And some of the work that you'll see today, you could execute in your own lab with that facility forthcoming. Right. So those are the goals for today. And I think before we really get into neuroimaging, and especially the complex methods we have to measure the brain, it's good to take a step back and just figure out why do we do neuroscience specifically. And really, these goals expand not only to neuroscience or brain science, but all sciences, right? And the goal really is to go from description to control, right? And so what does that mean, right? Well, people do things in the wild. We are all doing things every day, and we have descriptions of that, or the ethology of our behavior in the wild, from decades and really millennia of arts and literature, right? And those descriptions are useful, but what science is trying to advance is a level of explanation of linking brain models to our feelings, thoughts, and behaviors. And I would argue that really the first generation, the first era of human neuroimaging with MRI technologies did a lot of this explanation work, building models that linked brain structure and function to explain or correlate with our feelings, thoughts, and behaviors. Right. Now, if our models are any good, they might also not only explain, but also predict what people will feel, think, and do on different time scales, meaning predicting what people do before they even do it, second to second, or even kind of deterministically years in advance, right? Can we actually measure, and this is the whole idea with early biomarkers for any disease model, measuring things early in life to predict future life outcomes, all right? And I would argue this is sort of where the second era of neuroimaging, human neuroimaging especially, is trying to push the methods. And then lastly, if our models are really good, then we can control and manipulate and give us an opportunity to change and intervene to help people who have problems in these targeted brain circuits. Right. So description and control, and really my lab is trying to push explanation to prediction with our methods in humans, and a lot of the labs here at IQDAR are really actually doing the control aspect, right? You know which circuits to manipulate to give people motor control when they have new prosthetic limbs, for example. Great. So what is the ethology? What's the behavior that my lab cares about? Well, we somehow are able to stop ourselves when we're tempted by incentives, right? These could be incentives like tasty treats like marshmallows for your kids, but also you just imagine, walking trying to check out from Walmart, right? Just aisles and aisles of candy and sodas and chips tempting you as you're trying to leave the supermarket, right? And many of us sometimes are successful at not grabbing Funyuns on the way out of Walmart, right? But also a lot of times we fail to do those things, right? And there's obviously more insidious ways that we might fail to inhibit our impulses, and that includes pursuing drugs of abuse that can really lead to cycles of addiction. And so what we want to do is use the tools of neuroscience to study how we all vary in our ability to inhibit ourselves in the face of impulses, and even take inspiration from other mammalian species in which you can train dogs actually to do this, to inhibit their motor behavior, even with a tasty treat sitting right on top of their nose, until they do some kind of trick and then they can eat, right? So we want to take inspiration from other mammalian species, measure those circuits that allow these animals to do that basic inhibition of their impulses, and use it to study how we all vary in our ability to inhibit ourselves. So the way that an affective neuroscientist approaches this problem is by studying affect, or what we think of as the precursors to emotion. Right. And so the idea is, in this situation, there are cues in the environment, right? Tasty Funyuns or Snickers, that elicit neural activity in specific circuits, and you'll know what these brain regions are by the end of this talk. activity in those circuits generate what we call affect. So positive affect and arousal, or negative affect and arousal, or maybe more colloquially, excitement and anxiety. And those feelings are going to lead you to essentially approach or avoid these rewards, and some of these rewards might also be risky rewards, right? So going from affect, the feelings that your brain generates that bias your behavior towards or away from risky rewards. And we know a lot from the first generation of neuroimaging about the neural correlates of those affective states. Right? And so this is a meta-analysis of 27 studies by 2008 already, studying these circuits that are active when people anticipate gaining or losing rewards, in this case, just money. Right? And so these are two brain regions you're going to hear about a lot today, the nucleus accumbens, which we'll just call the NAC. This is a region deep in the brain that receives a lot of dopamine. And basically, activity in that region is more active when people are anticipating rewards. They haven't even received, the tasty treat or money. They're just anticipating that gain. In contrast, the anterior insula, anterior meaning towards the front of the brain, and the insula is this deep, insula is Latin for island. It's kind of tucked away deep in the brain as well. It's more active when people anticipate losing rewards. Right? So we already have this contrast of two circuits that might push you to approach or avoid incentives in the environment. And so my lab takes the approach of not only using functional imaging, but really going deep into the structure about how these circuits themselves are connected to each other. So our brain regions are all connected by white matter tracks, basically the wiring that allows brain regions to talk to each other. And what we do is characterize these targeted white matter connections that lead to the functional activity between those brain regions, right? Region A talking to region B, they're connected by a wire. We can measure both the structure and the function of that circuit. And then together, link structure to function to behavior like approach or avoidance of rewards. So this theme is going to recur throughout the talk, going from structure to function to behavior. And just sort of spoiler alert, this is the list of parts that we care about in the brain. And these are white matter tracks that converge on that NAC brain region. And we sort of tongue-in-cheek call it the connectome, because in neuroscience, connectomes, and really every ohm is popular right now. We don't think you have to measure everything in the brain. You just need to measure these handful of things that are relevant for motivated behaviors, right? And so these acronyms is gibberish to you right now, but we'll introduce to you to all these players throughout this hour. Great. So basically, how do you start doing this, right? Like, why am I telling you there's only 4 or 5 brain circuits that I care about measuring, right? Well, we and other neuroscientists do our homework, and homework for neuroscientists on humans is the old animal axon tracing literature, right? So that even dates back from the 60s and 70s. You can do your homework and basically figure out what is connected to what in the brain. And so these are studies from 97 and 2005 in monkeys that basically put a axon tracer in one brain region and sees where there is a monosynaptic or direct connection with other brain regions. And so these studies have established that there is a monosynaptic connection from the anterior insula to the nucleus accumbens, meaning the insula can talk to the nucleus accumbens. Now, the homework also told us that the NAC cannot talk back to the anterior insula. There are no axons that flow from that direction backwards. So basically, this is a unidirectional connection where only the anterior insula can talk to the NAC, but the NAC can't talk back. And so in putting this in more common parlance, right, if the insula is active for losses and the NAC is active for potential gains, then your loss circuit can talk to your gain circuit, but not vice versa, right? So that homework is sort of rich, right? Even from decades ago, can guide how we use human neuroimaging data to draw more deeper inferences. So how do we measure this connection in living humans, right? We obviously can't slice people's brains open to look for these connections and measure them. So we're fortunate to live in a time where this method, diffusion MRI, just another type of scan you can collect with an MRI scanner, allows us to actually see and measure those white matter connections, right? And so just briefly, we're measuring the diffusion of water molecules in the brain. And the idea is that water, right, if I threw a dot of dye into my water bottle, that molecule would diffuse isotropically, meaning evenly and randomly in all directions, right? So that's here on the left. Well, if you remember, I don't know, high school biology, you'll know that these axons, the wires that connect your brain regions, are wrapped in a fatty substance, myelin, which itself is a bilipid membrane, so water can't diffuse or permeate through it. So basically, your brain is inhibiting water from moving and in specific directions. So if we simply measure the jiggling of water in your brain, we can try to go backwards and model the white matter tracks that were inhibiting the water movement. All right, so anisotropic, not isotropic, or not even in random in all directions. So that's just the basics of diffusion MRI that allows us to look for things in the brain that are connected. And this is just some more details for aficionados out there. This is how we can do this type of method in every unique person's unique anatomy, neuroanatomy, right? All of us vary in our brain morphology, and this is the way we can target your brain in your specific brain space, rather than jiggling you into a template space. All right. So basically, one of the early wins from my work was basically, right, those papers from the 90s promised us that there was a connection between the insula and the neck. No one had ever visualized it in living humans, and with diffusion MRI, we were able to characterize that and see that connection from the anterior insula to the nucleus accumbens here in red. So this is just one of 30 plus subjects that we've been able to identify this connection, right? So sort of cool. You can see things in living human brains and non-invasively. Now, beyond that, we can measure the quality of that connection, right? And so there are many ways to quantify how strong those white matter connections are using diffusion MRI. But one of the most basic measurements that we can derive is a measure called fractional anisotropy. And the idea here is basically, on the left side, a value close to 0 means water is jiggling around that white matter track quite evenly and randomly in all directions. And so there's not much inhibition of the water. So that means that you might have maybe a less densely packed tract or less myelinated tract. And in contrast, the value closer to 1 suggests that, oh, water's moving in one direction relative to the two orthogonal directions. And that implies that there's a really robust white matter connection here inhibiting that water movement. So 1 means good. Good is and bad aren't the real terms we use, but maybe more coherent and less coherent tracks. Great. And so what we can do is actually derive that construct of really it's a metric of coherence along the trajectory of the connection, right? So from the insula to the neck, we can actually quantify how strong this connection is from the beginning to the end of the connection, right? And we can take this measurement and relate it to things like brain function and behavior. So great, we can see something that no one's seen before, and we can even measure the qualities of that connection. Now, the question now is, well, what do we want to relate it to, right? And I think this is a great example, a great way or time to use an example of the kinds of risk-taking and risky rewards that we can actually study and control in a lab setting, right? So I'm going to pose this question to you all. Here's a gamble that offers you a 25% chance to lose $5 and a 75% chance to win $1.75. How many of you would take this gamble if it's for real money? All right, we got Philip. We got 1 out of 40 people, 3 out of 40 people here, all right? All right, more people are trying to jump on the bandwagon, all right? Now, how about this gamble instead? Small probability, 25% chance to win $5, and instead 75% chance to lose $1.75. How many would take this gamble? All right, now you get pretty much the entire room, okay? So that difference between us all is already pretty interesting, right? And so what if I told you that both gambles are actually the same in expected value? Right? If you played both of these gambles even 10 times, we would all end up with $0, right? So why was it that only Philip initially liked this and then a few stragglers joined, like kind of reluctantly, but the entire room jumped at this opportunity here on the left, right? Well, this gamble on the left is positively skewed, meaning a small probability, well, it's a tail probability in the positive direction. Meaning, in this case, positive, meaning lots of money. Small chance to win lots of money. This one is a small chance to lose lots of money. It has a negative skew. And in the real world, positive skew gambles are popular, right? Just the popularity of casinos, right? We have a direct flight to Vegas and people love to gamble, right? It just makes sense. All the lottery games, all casino games, slots especially, highly positively skewed. Small probabilities to win lots of money, right? And what's sort of neat is is that the old school finance econ theories, they didn't really think about skewness. They only think about mean and variance. Mean meaning the expected value, and variance, the trial-to-trial volatility, which is actually equated between these two gambles. Mean and variance are also equated here, just the skewness differs. Anyhow, the question here is like these types of decisions that people make every day, can neuroscience help us explain part of that behavior? Right? And so years ago, we basically posed those gambles to people while scanning their brains with functional MRI. And the gambles that they saw were the same two that you all played, and also this symmetric gamble, which we use as a control because it's equated for mean and variance, but has no skewness, right? 50% win, 50% loss, there's no tail, right? And basically, long story short, with functional MRI, we can identify that the nucleus accumbens is more active before people even choose to gamble. And it's especially active for those positive skew gambles, those ones that you all jumped at the chance to play, just a minute ago, right? So we now have an interesting behavior and an interesting brain activity that precedes those risky choices. And in fancier modeling, we could do logistic regression to predict trial by trial whether people are going to gamble using activity in this circuit, and we can beat chance by a lot, actually. So with structure, function, and behavior, now we need to stitch these things together, right, as I've promised from the beginning. And what we then discovered was that people with a stronger connection from the insula to the neck, from the lost to the gain circuit, had lower preference for those gambles, right? So in all of you and all of us, how strong this connection is in one way dictates our preference for risky gambles like this. The stronger that connection is, the less you like those gambles. And beyond that, we can draw a link from that structural connection to neural activity in the nucleus accumbens, such that the stronger this connection is, the lower the activity in the nucleus accumbens when you're considering those gambles. And that in turn explains the gambling behavior, right? So we're going from structure to function to behavior. And we just found this to be really exciting, and we published it many years ago, and that's sort of the genesis of a lot of the research that my lab now does and applies to something like addiction. So before I proceed, I was curious if you had any questions about these methods. Yeah, Arnold?
The gambling behavior with a positive skill. Is there any correlation to this negative skill? And if it is, I feel that in a behavioral, if you have a less chance to lose, then it is a long game. So you're gonna gamble to get a bigger jump. In a negative skill, it's like a shot. Okay, I played for this much. I didn't lose. I didn't win. Then you stop and then.
Oh, I see what you're saying. Yeah, so you're thinking about this little more metacognitively than the average person thinks. Like no one knows. Oh, I got 30 trials. I should do it this way. And people don't know the structure of the experiment either. And beyond that, every trial is actually incentivized. They're making and losing real money on a trial by trial basis, right? So those decisions. Sorry, I'm just going to go backwards briefly. I think Arnold's suggesting that you might be able to do 8 of these, say no, and then say yes for the last 2. Like these kind of strategies, do people do that? And that's sort of the thinking that the economists would come back at us and make the claim, isn't there a higher level of rationality that people are imposing on this, right? And I think there's many ways to answer that. One is just ethologically, which is when we are all at the casino or at playing scratchers, you're feeling a certain way that's making you buy that scratcher for $1.50 at the gas station, right? And no one's thinking, I should play it every few months and in this order to do it. So I think the ethology there sort of grounds the phenomenon, which then allows us to use in a lab setting this situation and not overthink the way that subjects might be overthinking.
But is there any different correlation between negative skew versus positive skew?
No, and that's what's interesting. Yeah, this connection here, this is only true for the positive skew gambles. Right. So there's something about this, and I mean, you could relate this to foraging too. So naturally, lots of monkey and other animal species study the skewness preferences of animals who are foraging. Like, do they stay in one patch and then go to another where they don't know if there's going to be as much berries in that other patch, right? So same types of risky decisions, and positive skewness emerges in those foraging situations as well in other species. Right. Cool. I'm going to move forward. Right. So we want to basically, basically that work was the genesis of many other connections in the brain, and some of these brain regions you're probably familiar with the amygdala. You might be familiar with the prefrontal cortex, and especially this VTA region we're going to talk a lot about. We've basically been able to characterize more than just that insulated net connection over the years, and together we're calling that the connectome. Right? And so we want to deploy those methods on just pressing problems in society. And so two that I'll cover today is stimulant use disorder and how and relapse. So people who are trying to get off of drugs enter sometimes months long interventions, but once they leave hospitals, will relapse, especially in stimulant use disorder within 6 months. Right, and so despite the best efforts of clinicians and treatments, you'd like to maybe have a biomarker that while people are in your intervention, identify, oh, this person is highly likely to relapse, so that you can maybe be more flexible and target them with other interventions, right? And then lastly, developmental questions. So first of all, we just need to cover how dopamine even gets to the nucleus accumbens to increase that activity to make people approach risky rewards. Right? So this area, the ventral tegmental area, is a set of nuclei in the brainstem that produces the vast majority of dopamine for the brain, and through this green arrow projects that into the nucleus accumbens. Right? You've probably heard this called the mesolimbic dopaminergic projection, or just midbrain dopamine. This green arrow is what they're talking about in those podcasts and those kinds of settings. Right? Now, we know, thanks to rat studies combining functional MRI with optogenetics, basically turning on at will and off the VTA, that when you turn on the VTA, it's releasing dopamine in the nucleus accumbens and is actually responsible for the fMRI signal increasing in the nucleus accumbens. So this is causal evidence that we're not only drawing correlations between NAC activity and what we presumed was dopamine being released there. This is indeed causal evidence that turning on the VTA neurons to release dopamine in the NAC increases the NAC activity, fMRI activity. Right? And then in contrast, when you turn off those VTA neurons, you're getting less fMRI activity in the NAC. Right? So this is the VTA to NAC connection that we have also characterized and are now going to deploy in our studies of stimulant use disorder and relapse. So basically, long story short, in this more recent paper, we measured the white matter connections from the VTA to the NAC, shown here in gold. We also identified other things that were projected to other parts of the basal ganglia. The nucleus accumbens is part of a larger network of nuclei called the basal ganglia that control your motor control. And we really cared mostly about the VTA to NAC connection here in gold. And what we found was that in two data sets, both a discovery and a replication data set, is that people with a stronger VTA to NAC connection are less impulsive. Less impulsive in the sense of the trait impulsivity when you answer a bunch of self-report questionnaires that characterize your impulsivity. If you have a stronger dopaminergic projection, you're less impulsive by these self-report measures. Right? Beyond that, people with a stimulant use disorder, so these are people after chronic stimulant use, have a decremented or weaker VTA to NAC track. That's shown here by this lower, this dotted line is lower than the solid line, and so there's a weaker connection in people who have abused stimulants. And we can stitch all that together to show that, well, really, people with the stimulant use disorder, they report having high impulsivity, and they also apparently have these lower VTA to NAC connections. And our model can go from the fact that people have low VTA to NAC connections, are higher in impulsivity, and are thus more likely to have had a stimulant use disorder. Right? And here on the right is just showing that our classification of this beats chance and also beats clinical measures. Right? So beats clinicians using the standard self-report questions about a person's traits that might characterize stimulant use disorder. So that's neat, and this leads to the question, though: what, the chicken or the egg, right? Is this connection weak that precedes people trying drugs, or has that drug, chronic use of that drug, degraded those connections? And that's forecasting what is to come. So that was interesting, and we want to extend that to not only study, well, what's happening in the disorder. I've already shown you here on the left that the VTA to NAC connection is weaker in that disorder, But can we also predict who's going to relapse before they even leave the hospital, right? And this is where the research got interesting, because the VTA to NAC connection, that dopamine connection, could not predict who was going to relapse, right? We thought it would be really important, right? This is the actual throughput for how dopamine can get to the NAC. That could not predict who was going to relapse after they left the hospital. Instead, it's the insula to NAC connection that could predict, right? And that prediction was actually really interesting. It beat the self-report negative mood and even craving of drugs while they're at the hospital, and even the years of use of stimulants before coming to the intervention. So this is just showing our classification with the brain measurement beats those clinical measures. And we could even predict how many days until someone would relapse the moment they left the intervention, right? So this is a survival regression, basically predicting time until an event. And it's people with worse insula to NAC connections that relapse faster. So putting those two things together, essentially we have found that patients with a stimulant use disorder have a worse VTA to NAC tract connection. That midbrain dopamine projection is worse. That tract does not predict relapse, however. And instead is this other connection from the loss to gain circuits, the insula to NAC circuit, does predict the relapse, and actually the time, days until that occurs. Right? So this leads to the really tantalizing question is, right, we have all these circuits and they're not all doing the same thing, right? I think sometimes we're tempted to think of the brain as one, equipotential thing that can do everything. The point is we have targets in the brain and these circuits do different things. And so maybe the circuits that lead people to even try drugs are going to be different, right, not equal, to those degraded or altered by chronic drug use. And those also might be different than the circuits that are inhibiting you when you're trying to abstain and quit something that's addictive. And the only way we can now study that is to study the neural antecedents before even drug onset, right? And that's what leads us to this developmental question. Can we actually measure how this connectome develops structurally and functionally so that we can then predict before people even pursue drugs to begin with? So before I go to the last third of the talk, I'm just going to pause again for clarifying questions.
The last thing that relates to what you call control.
Right. When we start mapping colloquial words onto anatomy, you would think of the insular NAC as being like a control of what was generating the impulse to approach, which was the VTA NAC connection. Right.
For a better word, control.
Mm-hmm. What else do we have?
Integrative influence.
Sure. Yeah. I like that. Integration. Yeah. We normally think of the medial prefrontal cortex as integrating different values in the brain to create a subjective value. So that's a different connection altogether. Yeah, Delia?
We have a question from
Oh.
one of our Zoom participants. So did you observe any sex and age differences in your connection strength measurement?
Right. No, but we're underpowered statistically, and so this is sort of setting up the next study, which is right. This experiments we can do in small labs only has 30 to 40 usually healthy adults, or in this case, 40 healthy adults and another 40 patient population, like drug addicts. You need much more power to actually demonstrate sex differences, and that's something we are going to explore in the adolescents. We can even see how the difference of sex hormones that emerge during puberty lead to divergence in these connections. That might even bias, for example, male's propensity for externalizing disorders and female's propensity for internalizing disorders. That's going to be in part responsible, these tracks will be in part responsible for that sex difference. Thanks, very good question. Yeah, Avery? Right, in this case, oh yeah, good point. The question is, are maybe these people are all prescribed stimulants? Like, I mean, for example, what we call study drugs, things like Adderall and Ritalin are also stimulants. So in this case, these were not prescribed stimulants. All the subjects had an actual, I guess you would call it recreational substance use disorder. Yeah, good question. Last question, Pia?
Do you think this generalizes to other types of drugs? And then
Right.
is there any evidence for controlling for parent drug use?
Right, so comorbidity, right? I mean, most people who are abusing Stimulants are also reported, definitely nicotine addiction, sometimes also alcohol addiction. There's a lot of comorbid drug addiction. In this case, though, we focus on stimulants, first of all, because those drugs are dopamine agonists, meaning they increase dopamine in the brain. And we happen to know which brain circuits, like in this picture, create and receive dopamine. And so the effect of the drug is more targeted and less diffuse than, for example, alcohol, which influences the brain diffusely. So, I mean, that's my way of copying out and saying, well, this is the data set that we're studying and the handful of circuits that we know talk via dopamine, which is why we focus on stimulants. Good question about whether this will generalize to other drugs, though. Yeah, I don't know. All right, so let's try to tackle this other question, and this is really what my lab's been focused on the last couple of years. And really to set that up, the question is like, why are the kids not okay? Why do kids do crazy things, right? I don't have kids, so I don't really know, but I remember being kind of crazy when I was a kid. And basically, researchers have been theorizing about how the changes in the brain are producing that phenomenon, which we all see, right? Kids, may try risky things, right? They drive recklessly, right? They're cutting us off with massive trucks on Highway 49. Unsafe sex practices. They don't think about the consequences. They're trying drugs. They're doing riskier things. And so the explanation for that has normally come in two forms. One is we know that actually that some of these brain circuits are more and less active during adolescence. And we know specifically that the nucleus accumbens is actually more active, that gain circuit, and it peaks, that activity actually peaks is the highest at around age 15, right? So sophomore year of high school. In contrast, the loss circuit, we know that actually the anterior insula decreases during adolescence and hits a trough. Its nadir is at around age 16, 17. So basically, you have more activity for gains and less activity for losses, and that's one very clear explanation for why people are going after risky rewards. Secondly, people always talk about how, okay, these kids' brains are not fully formed, right? And that's an explanation for lots of things, including, kids can't rent a car until they're 25 because the insurance companies know that you got these reckless drivers. And the trite way to say it, there's lower connectivity from the front of the brain to other parts of the brain. Well, you now know some of the brain circuits that are actually truly not as well formed, especially that medial prefrontal cortex connection into the nucleus accumbens is the one that's not fully matured until the early mid-20s, right? And so this idea is that there's this low control or inhibition of those reward circuits. Right? Now, I like this third idea because this is, it's not counterintuitive, but it is at least going against the grain in the sense that maybe people pursue extreme exogenous drugs or rewards, may not only be drugs, other sensations out there. They might be doing so because their brain itself is not creating enough endogenous reward for the intrinsic rewards, like the joy of having done your chores for the day, or the joy of earning $5 of allowance for doing something at home, right? So basically, the intrinsic and endogenous reward that their brains are producing might just be blunted, and that's what's leading to more extreme exogenous sensation seeking, including drugs. So I like that explanation, and there actually is lots of recent evidence, from the mid-20 the 20-aughts onward, pursuing this idea, which is that it might be just blunted activity in the reward system for basic rewards like money and food that is leading to drug use. So that's exactly what this study found, and they measured with functional MRI the activity of those two circuits, the NAC and the VTA, when kids were anticipating rewards, and these rewards were simply points, winning 10 points or not winning 10 points, fictional points. And basically, lower activity in the VTA and the NAC when these kids were 14, when they're anticipating points, predicted 2 years later which kids would have actually a problematic drug use. You wouldn't call that a disorder at that point, but would be on the way towards like a diagnosable addiction. So already some evidence that for that third idea that it might be blunted activity that's leading to drug seeking. Right? And so basically that is where we come in with our methods, and We obviously don't have a magnet here yet. We will in a year, but we're fortunate to live in a time where not only the NIH, but many agencies are funding massive consortia, so multi-site studies, and making that data publicly available. Right? And so one of the major efforts is this Adolescent Brain Cognitive Development Study, ABCD, and they're following over 10,000 kids, starting at 9 years old for 10 years every year, and they're currently in that fourth year, so the kids are around 13, 14, right? And this is conducted at 21 sites across the United States, and the closest one to us would be in Tulsa at the Laureate Institute. And essentially, they're collecting not only these things, but they basically have thrown the kitchen sink of biomedical research at these kids every year. We care, though, about these handful of things. So every 2 years, they're collecting diffusion MRI so we can characterize the white matter tracks, and they're collecting functional MRI during that reward anticipation task. They're also collecting self-report impulsivity using this survey, and then every half year, and they call at the 6-month marks between the year in-person visits, they call and ask them about drug use, right? And so these are exactly the data that we care about and that we can use to answer interesting questions. Now, 13 is sort of young, right? And you're going to see in our data that basically, and it's probably a positive thing, not enough kids are trying drugs at 13. So we need to really extend our age range that we're studying. And so we're fortunate actually that the Imogen study did a similar thing, and have actually already completed this study in which they studied almost 2,000 kids across 8 sites in Europe and followed them from 14 to 22. And fortunately, they collected the same exact measures that we have available in the ABCD study. So we can essentially stitch those data sets together and get this age range from 9 all the way to 22. We can track the development of the connectome throughout that time and see if early measurements predict, and even predict maybe even onset, the year or the age that someone started trying drugs. Right. Great. And so I'm going to share with you some of the early results that we have. These are preliminary results, and I'm just curious on time, what we're looking like. Okay, cool. So, right, massive data sets. I think sometimes it's hard for people to appreciate that, going from studying 40 brains to studying 11,000 brains is a logistical warp jump. And that's also why it's great that IQBAR is going to have a whole technology and data architecture aspect. Because some of these massive data sets, just most people who are trained in small labs don't know how to use the kinds of computational resources for this type of data analysis. But our lab has done it, and basically, when we stitch together all the data from just the baseline when these kids are 9 years old, we have almost 200 terabytes of data. That's a terabyte is 1,000 gigabytes. So I don't know, about 10,000 of your laptop, right? So that's a lot of data. And right, it's kind of crazy that you go from 11,000 subjects down to really a fourth of that. And maybe some of you are thinking, like, when you were 9 years old, could you stay still for longer than a minute? Right? And so motion, basically, kids just wiggling in the scanner is a huge problem for data quality. And we're seeing this in a lot of the publications right now from this study, that you're throwing away a lot of data because the kids move too much and you have low quality data. And so that's also true for us. We went all the way down to just 2,500 data points in what I'm about to show you, right? And so just as a basic proof of concept, we were able to track these white matter connections, the connectome, in each and every one of those 2,500 kids and measure the qualities of those connections, right? So the VTA to NAT connection, that dopamine one, the insula to NAT connection, the amygdala to NAT connection, and also the medial prefrontal cortex, right? And so this is just the baseline measurement. And what's interesting is like the profile of these qualities are different than what we've previously published in adults. So there's already something that we're seeing different between 9 years old, obviously, and people in their 20s and 30s. And the question now is, with this data set, what's the trajectory of development from 9 to all the way to adulthood? We are able to replicate an old finding, which is that during puberty and at pubertal onset, actually, there is a change in the white matter near the insula. Right, so that's what Herding found back 12 years ago, and they did a method where they're just looking at the whole brain, looking at the difference between yes puberty and pre-puberty brains, and found specifically a change in white matter near the insula. Right, And so that sort of tipped our radar off to say, oh, what's near the insula? Well, the insula to NAC connection, right? So this specific lost to gained white matter circuit. And indeed, we're able to replicate that finding. And so this is white matter near the insula part of the tract, but it is part of this tract from the insula to NAC, the one that we have already explained can explain your risk preferences for those gambles, right? So it might be something about that circuit is specifically the one that's being altered at the onset of puberty. So that's structure, and really just a nice hint of things to come that even at the baseline level, we can just split the data from pre and post puberty and find this difference that was previously reported. Functionally, we can replicate early research just showing the increased activity in the mesolimbic dopamine system when people are anticipating winning money and also when they're actually gaining money. And I mean, this stuff might look nice, but this is nothing new for the last 20 years in neuroscience, so I'm just going to go fast. And essentially, we can draw activity out from those brain regions that we care about, the NAC, the VTA, the insula, and we can show you every 2 seconds, really, how the activity in these brain regions, whether they hit or miss, they actually win or lose the money, and for different conditions. And one of the things that we wanted to discover, which has actually not been found yet, is that the strength of the VTA to NAC connection should be related to NAC activity, right? If indeed that's the wiring that allows the VTA to release dopamine into the NAC, then a stronger structural connection should relate to greater NAC activity, right? And that's actually what we were able to find with this data set. It's very exciting to see that the strength of the VTA to NAC structural connection correlates with greater NAC activity during reward anticipation. And it's true for both your left and right hemisphere. You have two hemispheres in your brain, and it's true for both hemispheres. So again, an early functional finding. And really trying to ask the hard question, which is, can we take those measurements at age 9 and predict, this time it was 3 years follow-up, so 12 years old, predict future real-life drug seeking. And at that time point, this was basically this is 3% of the data set. Only 48 had tried a recreational drug. I think that's a positive thing, right? It's kind of morbid to want a larger sample in this case, but that's what we were working with. And what we were able to find was indeed a worse VTA to NAC connection at 9 years old did predict that these kids at 12 years old would have tried a recreational drug, right? Oh, whoops. And then finally, beyond that, we can link the track to the trait impulsivity, as we've done before, to that future drug use. Is the video okay with what I just pressed? Okay, great. Okay, so that is a replication and extension of what I just talked to you about earlier, which is that in adults who had abused a stimulant use, a stimulant drug, had also the same pattern of results. But this is showing you that those problems might precede drug onset and not be a result of drug abuse. Right? So this is an example of answering that these tracks are antecedent to drug seeking, not a result of chronic drug use. Right. So exciting things. And lastly, this converges with that earlier study from 2016 that showed that it's really that third idea. Blunted NAC activity for endogenous rewards is predicting drug use. And this really fits nicely into that, we wouldn't call it a theory, but that conjecture. Great. Okay, so finally, zooming out, these projects, we've been very fortunate to be funded to explore this with the Arkansas Biosciences Institute support. And right now, we're under review with a center proposal with researchers at UMS in Little Rock and the Arkansas Children's Research Institute. And together, putting many projects and research labs together, trying to form a center for studying the developing brain. And this is like, we're just at the early days of doing this work, but you can now see mapped out what you've seen today, mapped up across time for this whole decade of life. And then lastly, we also collect data on this other really tantalizing idea that, in adulthood, how much can you change, right? And so we are actually testing the extent of structural plasticity in these connectome circuits in adults, so 25 and older. And this is a study that we have completed data collection at the Laureate Institute in Tulsa. So that's zooming out of this study specifically, but lastly, I just want to zoom out to what the future of MRI research might look like at that forthcoming magnet in IQDAR. And so this is an example of the faculty across 11 different departments on our campus and the different research areas they have that we stitched together in a proposal to the NSF to help fund the magnet that we're going to purchase. And so, what I've described to you is just one small nugget, really one brick in a wall that really represents a large community and a growing community of people who can ask interesting questions, research questions with MRI. So, summarizing for today, hopefully I have answered these three things, which is we do our homework, use different MRI scans to measure the structure and function of reward circuits in humans. We can actually use these measurements to study stimulant drug addiction and show that there are different circuits that might be relevant for different phases of the disorder. And then tentatively, yes, we can actually use early measurement of these circuits to predict future drug use, right? And this is, again, setting the stage for what's going to be really exciting coming out of IQDAR in the coming years. So I just want to thank the funding and my collaborators, and this is how you can actually, with the code, do this at home. All right, thanks, and I'll be here for questions. Yeah, Jimmy.
Thank you. That was great. Enjoyed it and learned a lot. I'm curious, right at the end, we talked about, well, maybe we could look at promoting plasticity in adults to sort of target this structural deficit, I guess, a structural condition in the brain. And I'm curious, what kinds of things are people, you and others, thinking about, and then would they be actually potentially more effective if you implement that in adolescents?
Right, just a big question about plasticity, like just one of the gold standards that we're all trying to tackle. And it's been plasticity later in life has been demonstrated in other circuits, obviously, like learning new motor skills and in cases of stroke and in memory circuits. In this case, right, your preference for different rewards like Snickers or Kit Kat, right? That's kind of fixed. I feel like what you liked as a kid, you're still buying at Walmart 20 years later. And so if we can show plasticity of those deep reward circuits and the preferences that emerge from that is a very powerful demonstration. And the question is how to do it, right? And that's unanswered. We're not trying this, but one possibility, the two real more invasive possibilities, one is stimulating specific circuits. What if you stimulated the MPFC to NAC in a specific pattern that leads to long-term potentiation that would then lead to greater myelination of that specific tract, right? Obviously cannot do that in healthy humans. So stimulating specific circuits and seeing that effect on the white matter plasticity. Second might be neurofeedback, right? Just training people on increasing or decreasing their own brain activity using, basically, you can show people a thermometer of activity in their own brain region while scanning their brains and say, hey, try to increase activity or try to decrease. So in a way, developing your own control over your brain with the feedback of your own brain activity. That would be another way to do it, not less invasively. But it really is an open question. And one way I'm thinking about using the adolescent data to answer this, like what's the extent of structural plasticity, is so towards the end of adolescence, towards early adulthood, right? You presumably have this tapering of plasticity. And so if we take the influence of functional activity in that reward task at, say, like 19 years old, and see how much that relates to structural changes at 22 years old, like in that European data set, that sort of sets an upper bound of how much plasticity one could expect from a basic experience, like gambling for money. So that's the way I'm thinking about using that data set in adolescence, to set an upper bound for what we could expect in adults in their, I'm an old guy in my 30s, like how much can my brain possibly change if these 22-year-olds have that much change from experience? Right. Yeah, in the back. Right, absolutely, yes. Right, yeah. So this is a question about, which is another focus of IQDAR. How can we integrate what we know from nutrition and food science, which is the strength of this university, actually, and actually agricultural insights, leverage that insight to harness that to enforce neuroplasticity. Yeah, that feels like a rich area that our institute is primed to try. Risky though, because I mean, some of these things you don't know how long of a diet would you need to see a measurable change with some of these crude MRI methods. But I love the idea. Yeah, thank you, Michaela.
Do you have any predictions about, I talk about this all the time, so bear with me, but contraceptives? There's some evidence that show that women who use contraceptives are more prone to alcohol abuse because of the depressant effect, and they have like less GABAergic signaling. So I know you were focused primarily on stimulant use, which is obviously kind of the opposite of this, but would you have any predictions about like tracks that might be involved in that process for women who are using them?
All right, so GABAergic projections. I want to go back to the picture of this connectome. We know that all these circuits, these three circuits from the front of the brain and also from the amygdala to the neck, are glutamatergic, so they're presumably able to excite the neck. And the V theta NAC connection is dopaminergic. Now, the GABAergic signaling usually happens through the interneurons that are basically sitting next to the neck, and those, I mean, they're very interesting. Like, how does this all play together? You have circuits saying yes, other circuits saying no. How do they all play together to lead to any behavior at all? And I don't have an answer for the GABAergic projections that are maybe influenced by hormonal contraceptives, but that's, you know, in this scheme, that's also going to play a role. Yeah, Jeff.
So in your research, do you ever factor in what I'd call family backgrounds versus, say, multi-generational factors of abuse for, say, drug or alcohol versus ones that aren't? And compare that to your data?
Thanks for asking that, because in the ad for this talk, I gave a quote about family history of drug use, and I haven't even talked about this. So one of the greatest risk factors and greatest predictors of a person themselves getting a substance use disorder is having a family history of substance use disorder. And we're talking 4 to 8-fold increased risk if you're simply had a biological person in your family tree that had their own substance use disorder. So that's actually is part of the grant that we're pitching with the COBRI, which is both data sets have collected family history use data. And the question is, how is that even early in life biasing the development of these circuits by 9, right? In some cases, some people think 9 is too old to be studying kids. I think that's ridiculous, studying adults my entire life. And some people are even doing MRI fetally to look at really early impact of something like family history and genetic effects, right? So that is actually one of the questions we have, which is what's happening here as a result of family history of drug problems, and also what's independent of that. So I don't have an answer for you yet, but next year at the fancy building, I'll tell you, Jeff. Yeah.
On that same line, with just different addiction in like family history, like, is there a link that you've seen or any other research has seen between like drug addiction versus alcohol addiction versus gambling addiction, and if they correlate or if there's pretty differences in each in terms of family history and stuff?
Right. Yeah. So other risk-taking behaviors. So ever since we found that first connection of INSA to NAC, it's really fun to see how other researchers are applying that measurement in their own fields of interest. And there is a lot of interest in gambling disorders, especially with more like sports gambling getting more and more legalized throughout the nation. So I imagine in the coming years we'll see a lot of work on this specific circuit and its relation to gambling disorders. Right. Yeah. Thanks for that question.
Curiosity, for the families of these children, are you aware of socioeconomic status?
Yes, we have that measurement, and we always use it as a control variable of no interest. So how would you use it as a variable of interest?
I'm just curious as a
Right.
so
Right.
how they
Summarize for.
Right. The question is about socioeconomic status and how do those studies measure it? The answer is yes, and usually in crude ways, like years of education and family income, right? Ways that don't really capture the nuance of social and economic factors in one's life, right? Anyhow, and I was making a quip that we normally in neuroimaging use those as variables to throw out as noise and show that our measurements matter more than that. But there's a lot of potential for studying the actual socioeconomic factors that lead to drug use. Right. Are we good on time, or should I keep going?
Well, it is 10, so we can keep it going. Anybody that's in the audience have more questions, and we have the live stream up, and we let them know.
Great.
But it's up to you.
Yeah, let's get Justin, and then I'll be here.
Have you explored other techniques for looking at brain structure and functioning, or is it mainly the MRI?
Yeah, I have not, but are you thinking like FNIRS and EEG, which will also be available?
Yeah, I was just kind of curious if you have looked into this
Right.
before.
I have not. The issue with both of those methods, actually, they're surface methods, right? You can actually measure deeper into these limbic circuits, right? Yeah.
I was thinking about trauma and maybe the opposite of trauma, something very meaningful and positive.
Right.
Either one could trigger the dopamine energy pathways toward forming long-term memories embedded in the campus.
Right.
And you speculate on those kind of words.
Right. So just pleasurable memories. How do we form them? If, like, your the first gift your grandma ever gave you for Christmas, like these kinds of things, like really, makes me happy. So people study that. There are dopamine projections from the VTA to the hippocampus. Very active area of research. People do optogenetics to actually control and implant like false memories, but make them pleasurable. These kinds of things. Hippocampus also does connect into the basal ganglia, and so that's actually an unanswered question about how exactly you form these these formative memories when you're young. And maybe you're right. I should add a circle for the hippocampus and study its projections into the striatum. Thank you for the idea. All right, great. Maybe we should wrap it up so that people can, finally have breakfast, and then, and I'll be here, obviously, field more questions. So thank you for your time.